Welcome to the deep dive. Okay, so you're starting your medical journey, maybe first year, and wow, the amount of information, it can feel like a tidal wave, right? Totally overwhelming sometimes. So today, we want to cut through some of that, give you a foundational starter pack, if you will, for understanding infection. It's really crucial stuff. Yeah, think of this as your bedrock knowledge, our mission, to kind of navigate this incredibly diverse world of microorganisms. We're talking everything from, tiny bacteria all the way to these, mysterious things called prions. It's like meeting the cast of characters you'll be dealing with your whole career. And we're drawing on reliable medical sources here, but presenting it with hopefully some useful mental anchors, metaphors, maybe some acronyms just to help it stick. Exactly. We're building a mental framework, not just listing facts. So as we get started in this deep dive, here's a little something to chew on, a sort of pre-flight check question. What fundamentally makes something living in biology? Keep that in the back of your mind because some of the things we'll meet today really stretch that definition.
That's a perfect place to start because what we're diving into is, it's a huge spectrum of organisms and agents linked to infection. You've got everything from complex, multi-celled creatures down to things that honestly aren't technically alive but still cause major problems. So when we talk about this microbial cast, we can sort of break them down into two main buckets. First, the clearly living microorganisms. These are the ones you probably expect. Bacteria, archaea, fungi, helminths. Those are parasitic worms, protozoa. Even some algae fit in here. And then you have the ones that are, not technically microorganisms, but they are definitely pathogens. That's viruses and prions. These are rule breakers. We'll definitely circle back to them because they really make us question that what is life definition. Okay, so to help anchor all this, let's use a metaphor. Think of a cellular city. The biggest division in this city is based on cell structure. Prokaryotes versus eukaryotes. Let's start with prokaryotes. Think of these as the studio apartments of the cellular world. Basically, they're unicellular, just one room. They don't have a true membrane-bound nucleus. They're genetic material. It just kind of floats around in a region called the nucleoid. And crucially, they lack other membrane-bound organelles like mitochondria or an ER. Simple, efficient, they get the job done. The main examples you'll deal with are bacteria and archaea.
Archaea. You hear about them, but maybe less in a clinical context.
Exactly. Archaea are fascinating prokaryotes, often found in really extreme environments, think boiling hot springs or super salty lakes. But for medical students, the key thing is they generally aren't considered direct human pathogens. So our focus, clinically speaking, is almost always on bacteria.
Gotcha. So bacteria are the main prokaryotic players we need to know. What about the other side of the city? The eukaryotes.
The eukaryotes, these are the mansions. They have a true nucleus, like a master suite, where the genetic material is neatly contained. Plus, they have all sorts of other membrane-bound organelles, mitochondria for energy, endoplasmic reticulum for protein synthesis, you name it. Each room has a specific function. This complexity allows them to be, generally larger and capable of more specialized tasks. And the pathogens in this group, they include fungi, algae, protozoa, helminths, and other parasites.
So studio apartments versus mansions, it sounds like more than just looks, right? These differences must really matter functionally?
Oh, absolutely. Huge functional and clinical implications. How these different cell types operate, how they cause disease, and critically for you, how we treat the infections they cause. It all ties back to these fundamental structural differences. Treating a prokaryote infection often involves targeting things they have that our eukaryotic cells don't.
Okay, that makes sense. So let's zoom in on our first major player then. Bacteria. These prokaryotic studio dwellers. They're unicellular, found pretty much everywhere. Soil, water, on us, in us. And we need to get used to their names, right? That binomial nomenclature.
Yes, genus and species, like Staphylococcus aureus, often just shortened to the S. laureus in notes. You'll see that constantly.
Okay, so how do we start classifying them? It feels like there must be thousands.
There are. But to get you started, especially when looking at initial lab reports, think SSA. It's a handy anchor: shape, staining, and air.
SSA. Okay, I like that. Let's break it down. First S, shape.
Right, morphology. How they physically look under a microscope. There are three basic shapes you'll see all the time. First, koshi. These are spherical, like little balls or berries. Think Staphylococcus aureus, common cause of skin infections, or Streptococcus pneumonia, a big player in pneumonia.
Kochi spheres. Got it.
Second, bacilli. These are rod-shaped, like tiny sausages or pills. Classic examples are Escherichia coli, or E. coli, you hear about it with food poisoning, and Bacillus anthracis, which causes anthrax.
Bacilli rods, okay.
And third, spiral shapes. This is a bit more varied. It includes things called spirilla, Vibrio, spirochetes. They can look like corkscrews or commas. Treponema pallidum, the bug that causes syphilis, is a spirophase. Helicobacter pylori, linked to stomach ulcers, is another spiral type.
Does the shape actually affect how they cause disease?
It can, yeah. Sometimes shape influences how they move, maybe how they stick to surfaces inside the body, or even how they avoid our immune cells. That corkscrew shape of a spirochete, for example, might help it burrow through tissues. So shape isn't just descriptive, it can hint at function.
Interesting. Okay, so that's the first S shape. What's the second S in SSA?
The second S is for staining properties, and specifically the Gram stain. Honestly, this is a cornerstone of diagnostic microbiology, a real game changer.
The Gram stain. Heard of it? Definitely sounds important. How does it work?
The principle is pretty neat. You use a series of dyes, crystal violet, then iodine, then an alcohol wash, and finally a counterstain called safranin. These dyes interact differently with the structure of the bacterial cell wall, and this gives us two major groups: gram-positive bacteria and gram-negative bacteria. Gram-positives have a really thick peptidoglycan layer in their cell wall. Peptidoglycan is this strong, mesh-like molecule, but crucially, they don't have an outer membrane, and they don't have something called lipopolysaccharide, or LPS. Because that thick peptidoglycan wall holds onto the crystal violet iodine complex even after the alcohol wash, they appear purple under the microscope. Our anchor here is simple: purple for positive. Think Staph aureus again, or Streptococcus.
Purple positive. Okay, so gram negatives must be different.
Exactly. Gram-negative bacteria have a much thinner peptidoglycan layer, but, and this is a huge difference, they do have an outer membrane outside that thin layer, and embedded in that outer membrane is LPS, lipopolysaccharide.
LPS, you mentioned that. Why is it significant?
LPS is also known as endotoxin. When gram-negative bacteria die and break apart, this LPS is released, and it can trigger a massive inflammatory response in us, the host. It's a major cause of sepsis and septic shock. So that outer membrane with LPS makes gram-negative infections potentially very dangerous and require specific treatment approaches.
Wow, okay, so structurally different, and that LPS is bad news. How do they look on the stain then?
Because their thin peptidoglycan layer and outer membrane don't hold on to the crystal violet iodine well, the alcohol wash removes the purple dye. Then they pick up the pinkered counterstain, safranin, so they appear pink or red. The anchor, pink for negative, think E. coli or salmonella.
Purple for positive, pink for negative. Got it. And you said the staining's a game changer. Why is it about treatment?
Absolutely. This is often the very first piece of critical information a doctor gets back from the lab. That difference in cell wall structure, thick peptidoglycan versus thin layer plus outer membrane, means that different antibiotics work better against gram-positives versus gram-negatives.
So knowing purple or pink helps choose the right weapon, essentially.
Precisely. Often in serious infections, you might start an empirical antibiotic based just on the gram stain result and the clinical picture, even before you know the exact species. It buys critical time.
Yeah.
So yeah, understanding gram-positive versus negative is fundamental for antibiotic selection.
Okay, that makes total sense. So shape, staining, the final A in SSA is for air, right? Oxygen needs.
That's it. Oxygen requirements. Not all bacteria breathe the same way. You have aerobic bacteria. They need oxygen to grow, just like us. Mycobacterium tuberculosis, the TB bug, is a classic example. Loves the oxygen-rich environment of the lungs.
Aerobic need oxygen. Makes sense.
Then you have anaerobic bacteria. They thrive without oxygen. In fact, oxygen can be toxic to some strict anaerobes. Clostridium botulinum, which causes botulism, often grows in canned foods where there's no oxygen.
Anaerobic, no oxygen.
And then there are the adaptable ones, facultative anaerobes. They're metabolically flexible. They can use oxygen if it's around, but they can also switch to anaerobic respiration or fermentation if oxygen isn't available. Many common pathogens fall here, like E. coli and Staphylococcus aureus. They can grow in lots of different body sites.
Okay. SSA, shape, staining, air. That's a really helpful framework for starting to sort out bacteria. Now, how do they grow? You hear about bacterial infections spreading fast.
They do grow fast, typically by binary fission. One cell just splits into two identical daughter cells. Two become four, four become eight. It's exponential or logarithmic growth under ideal conditions.
And this growth follows a pattern.
It does, a predictable curve. We can remember the phases with the acronym lag-log-stat-def.
Lag-log-stat-def. Okay, let's unpack that marathon.
Right. The lag phase is the warm-up. Bacteria are adapting to the new environment, making enzymes, getting ready, but not actively dividing much yet.
Just getting settled in.
Exactly. Then comes the log phase, short for logarithmic. This is peak performance, rapid cell division, exponential increase in numbers. This is often when disease symptoms are most severe and also when bacteria are most susceptible to antibiotics that target growth.
Log phase is the boom time.
Yeah.
Then what?
Then they hit the stationary phase, kind of like hitting a wall. Resources like nutrients start running low and waste products build up. So the rate of new cell growth equals the rate of cell death. The population size levels off.
Makes sense. Resources aren't infinite.
Right. And finally, if conditions don't improve, you get the death phase. The collapse. The number of dying cells exceeds the number of new cells, and the viable population declines. It's like a whole life cycle for the population in that culture tube or infection site.
That's a great way to visualize it. You also mentioned something about some bacteria forming.
Yeah.
Endospores. What's that about?
Ah, yes. Some bacteria, like Bacillus and Clostridium species, can form endospores when conditions get tough, like lack of nutrients or water. These aren't reproductive structures, they're dormant survival pods. They basically shut down metabolism, dehydrate themselves, and form a super tough protective coat. This lets them survive incredible stresses, boiling, radiation, disinfectants, for potentially years. When conditions get better, they can germinate back into active vegetative cells.
Wow. So that must be a huge problem in hospitals, right, for sterilization?
A massive challenge. Standard disinfection might kill the active bacteria, but not the endospores. You need harsher methods like autoclaving high-pressure steam or specific sporicidal chemicals to ensure you kill them. They're incredibly resistant.
Okay, so bacteria prokaryotes, SSA classification, lag-log stat death growth. Some have these tough endospores. We explored the studio apartments. Now let's move to the mansions, the eukaryotes. You mentioned fungi are key players here.
Indeed. Fungi are eukaryotes, so structurally they're more like our own cells, which, as we'll discuss, has implications for treatment. You often hear about them causing opportunistic infections.
Meaning they take advantage of a weakened host.
Exactly. Someone whose immune system is compromised, perhaps due to illness like HIV, or medications like chemotherapy or immunosuppressants after a transplant. Fungi that might not bother a healthy person can cause serious disease in these individuals.
What forms do fungi take? Are they all like mushrooms?
Yeah, not the ones causing infections usually. Medically relevant fungi mainly come in two forms. First, yeast. These are unicellular, usually round or oval. They reproduce by budding, mostly. A classic example you absolutely need to know is Candida albicans.
Candida causes thrush, right? And yeast infections.
That's the one. Oral thrush. Vaginal candidiasis. Skin infections. Very common. The other main form is molds. These are multicellular, forming filamentous structures called hyphae. These hyphae grow and branch, forming a network called a mycelium. Think of Aspergillus fumigatus, a mold that can cause lung infections, especially in immunocompromised patients.
Yeast or single cells, molds or filaments. Got it. What kind of diseases do fungi cause overall?
A whole range. You have common superficial stuff like ringworm and athlete's foot. Then there are more serious lung infections like fungal pneumonia, the candidiasis we mentioned, and aspergillosis. We often categorize them by how deep they go. Superficial mycoses stay on the very surface: skin, hair, nails. Subcutaneous mycoses get into the tissues just under the skin. And the most dangerous are systemic mycoses, where the fungus spreads to internal organs. These are the ones most often seen in severely immunocompromised patients.
Okay, fungi eukaryotes, yeast, and mold forms, often opportunistic, causing anything from skin issues to systemic disease. Which leads us nicely to our next question for you listeners. Thinking back to bacteria and now fungi, why might treating fungal infections be trickier than treating bacterial ones? Keep that thought simmering. For now, let's quickly cover a few other groups involved in infections. We use the SSA for bacteria. Maybe we can use the W's and the A's for these. Worms, wiggle cells, and arthropods.
Ha! I like that. Okay, the first W worms, or helminths, these are multicellular parasitic worms. Think tapeworms, hookworms, pinworms. They're eukaryotes and often quite large compared to bacteria or fungi. You don't usually call them microbes because you can often see them. Their life cycles can be really complex, sometimes involving different hosts or stages within the human body. Diagnosis often involves finding eggs or larvae in samples like stool.
Okay, helminths are the worms. What of the wiggle cells?
That would be the protozoa. These are single-celled eukaryotes, so still mansions in our city analogy, but just one mansion. Many are important pathogens. Think Plasmodium, the parasite that causes malaria, or Trichomonas vaginalis, a common cause of sexually transmitted infection, or Toxoplasma gondii, causing toxoplasmosis.
So single-celled eukaryotes different from bacteria.
Right. They often have complex life stages, too, like motile forms that cause disease and dormant cis forms that help them survive outside a host and spread.
Okay, worms and wiggle cells. That leaves the A, arthropods, like insects.
Exactly. Arthropods, insects like mosquitoes and fleas, arachnids like ticks and mites. Their main role in infectious disease usually isn't causing the disease themselves, but acting as vectors. They're the disease delivery service.
Vectors, meaning they transmit other pathogens.
Precisely. Mosquitoes transmit malaria, caused by a protozoan, and dengue fever, caused by a virus. Ticks transmit Lyme disease, caused by a bacterium. Fleas can transmit plague, also bacterial. Understanding the vector's life cycle and habits is absolutely critical for controlling these diseases.
Wow, so controlling the delivery service can be as important as fighting the pathogen itself. Okay, so we've covered bacteria, fungi, helminths, protozoa, and the arthropod vectors. These are mostly, well, living in the traditional sense.
Right. But now let's circle back to that question we started with. What defines living? Because our next two groups, viruses and prions, really push the boundaries.
The rule breakers. Let's start with viruses. Why aren't they considered alive?
Okay, viruses. The obligate hijackers. The core reason they're not typically classified as living is that they lack the machinery for self-replication and metabolism. They're basically just genetic material, either DNA or RNA, wrapped in a protein coat called a capsid. Some also have an outer fatty layer called an envelope. But critically, they are obligate intracellular parasites. They must get inside a living host cell to replicate. They can't grow, they can't make energy, they can't reproduce on their own. They hijack the host cell's machinery to make copies of themselves. That lack of independent function is key.
So they need us, or other living cells, to make more viruses. How are they classified?
Mainly by their genetic material, DNA viruses versus RNA viruses, and also by whether they have that outer lipid envelope enveloped or not, non-enveloped. Examples you'll definitely encounter. DNA viruses include herpes simplex virus, varicella zoster, causes chickenpox shingles. Hepatitis B RNA viruses include HIV, influenza, rhinoviruses, common cold, and coronaviruses.
Okay, and how do they actually do the hijacking? Is there a pattern?
There is a general cycle, yeah. We can use the acronym AERAR to remember the steps. Attachment, entry, replication, assembly, release.
AERAR.
Attachment. The virus binds to specific receptor molecules on the surface of the host cell, like a key fitting into a lock. Entry. The virus or its genetic material gets inside the cell. This can happen through fusion with the cell membrane or by being taken in through endocytosis. Replication. This is the hijacking part. The virus uses the host cell's enzymes, ribosomes, energy, everything to copy its own genetic material and produce viral proteins. Assembly. The newly made viral components, genetic material, and proteins come together to form new virus particles, or virions. Release. The new virions get out of the host cell, often by bursting the cell open, lysis, or sometimes by budding off the cell membrane ready to infect neighboring cells.
It's like a microscopic hostile takeover.
Okay.
So viruses aren't alive because they need a host cell, but then prions, what are they? They sound even stranger.
They are arguably the strangest. Prions are the misfolded menace, and if viruses stretch the definition of life, prions seem to completely defy it. Why? Because they are simply misfolded protists.
It's proteins, no DNA or RNA at all.
None whatsoever. No genetic material. That's the mind-blowing part. They are infectious agents composed solely of protein, so they can't replicate in the traditional sense. They don't grow, they don't metabolize, they fail pretty much every standard test for life.
But they cause disease. How?
They cause devastating neurodegenerative diseases called transmissible spongiform encephalopathies, or TSEs. The best-known human one is Creutzfeldt-Jakob disease, CJD. The spongiform part refers to the way the brain develops holes looking like a sponge due to neuron death. The mechanism is, well, unique and frankly terrifying. We all have normal prion protein, PRPC, in our brains, which are thought to have some function, though it's not fully understood. They have a normal shape, mostly alpha helices. An infectious prion, PRPSC, is the same protein, but it's misfolded into a different shape, rich in beta-pleated sheets. When this misfolded prion encounters a normal prion protein, it somehow induces the normal protein to refold into the abnormal pathogenic shape.
So it converts normal proteins into bad ones, like a chain reaction.
Exactly like a chain reaction. These misfolded proteins then aggregate, forming clumps or plaques in the brain that disrupt nerve cell function and eventually kill the neurons.
That's unsettling. And are they hard to deal with?
Incredibly. Prions are highly resistant to heat, radiation, and many chemical disinfectants that would destroy viruses or bacteria. And because they are technically just altered versions of our own proteins, they don't trigger an immune response. No inflammation, no antibodies. Our body doesn't recognize them as foreign invaders.
Wow. No genetic material.
Yeah.
Resistant to killing, invisible to the immune system. It's truly a unique challenge. Okay, so that brings us back to the question we parked earlier. Based on everything we've discussed, especially the eukaryote-prokaryote difference, why is treating fungal infections often harder than bacterial ones?
Right, let's connect the dots. Remember our cellular city. Bacteria are prokaryotes, studio apartments, while fungi and humans are eukaryotes, mansions. Because fungal cells are eukaryotic, like our own cells, their fundamental machinery, ribosomes, metabolic pathways, cell membrane components are much more similar to ours compared to bacteria. This makes it really challenging to find drugs that kill the fungus without harming human cells. Selective toxicity is harder to achieve. Antibacterial drugs often target things unique to prokaryotes, like peptide-glycan cell walls or different ribosome structures, which eukaryotes don't have. Antifungal drugs have fewer unique targets. They might target ergosterol, a lipid in fungal cell membranes that's different from our cholesterol, or the fungal cell wall, which is different from bacterial peptide-glycan. But because of the underlying similarity, there's often a narrower therapeutic window and potentially more side effects with antifungals.
Ah, the mansion similarity makes them harder targets without causing collateral damage to our own mansions.
Well,
That makes perfect sense. So there you have it. That was quite a tour of the microbial world, wasn't it? From prokaryotic bacteria with their SSA and growth cycles to eukaryotic fungi, helminths, and protozoa, plus the arthropod vectors.
And finally, the rule-breaking viruses and truly bizarre prions that challenge our very definition of life.
Exactly. And mastering these differences, understanding these categories, it's not just for exams. It's genuinely fundamental for clinical practice, for diagnosis, for treatment decisions you'll make every day. These are the building blocks.
Couldn't agree more. This foundation is crucial.
So as we wrap up this deep dive, here's a final thought to take with you. These infectious agents aren't static, right? They evolve, they adapt. How do you think this constant evolution, especially with their unique biological traits like viral replication or prion conversion, will continue to challenge medicine? How might our strategies for prevention and treatment need to change in the face of these ever-evolving threats? Something to ponder as you continue your journey. Thanks for joining us on the Deep Dive.